Participants join us in different parts of the world. Assalamu alaikum and very good morning according to your country and your time zone. My name is Gauthier Rocco. I am program manager at Comstech, and it is my pleasure to serve as moderator for today's online training workshop on quality assurance and physicochemical analysis of drug formulations organized by OIC Comstech. Today's workshop focuses on an important area within healthcare and pharmaceutical sciences. Ensuring the quality control system are essential for protecting public health, strengthening regulatory frameworks, and building confidence in pharmaceutical products. In this context, the objective of this training is to enhance technical knowledge and practical skills in pharmaceutical quality assurance, physicochemical analysis, and regulatory practice, while promoting knowledge exchange and collaboration among pharmaceutical professionals, regulatory authorities, researchers, and academic institutions across OIC member and observer states. We are honored to have our distinguished resource persons with us today, and we look forward to their valued contribution and fruitful discussion through this program. Before we proceed with the opening remarks, we will begin with the recitation of Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الرحمن علم القرآن خلق الإنسان علمه البيان الشمس والقمر بحسبان والنجم والشجر يسجدان والسماء رفعها ووضع الميزان ألا تطغوا في الميزان وأقيموا الوزن بالقسط ولا تخسروا الميزان صدق الله العظيم.
Thank you. We will now proceed with the welcome address by His Excellency Professor Dr. Mohammed Iqbal Chaudhry, Coordinator General of OIC Comstech. As His Excellency is currently traveling outside of country, he has kindly shared his opening remark through a recorded video. We are kindly...
Bismillahirrahmanirrahim. Distinguished speakers, dear participants, colleagues, ladies and gentlemen, Assalamu alaikum and very good afternoon. It gives me a great pleasure to welcome you all to this online training workshop on quality assurance and physicochemical analysis of drug formulation organized by OIC Comstech. The availability of safe, effective and quality medicine is one of the fundamental requirements of any healthcare system. Yet many countries continue to face challenges related to medicine quality, regulatory framework, laboratory infrastructure, and the growing circulation of substandard and falsified products. Addressing these challenges requires not only stronger regulatory systems, but also continuous capacity building and knowledge sharing among professionals working in the pharmaceutical sector. Ladies and gentlemen, this workshop has been designed with the objective in mind. Through the expertise of our distinguished speakers, participants will gain insight into quality assurance of pharmaceutical products, analytical approaches used in quality control laboratories, and the regulation and quality assessment of traditional medicine. These are areas of increasing importance for many OIC member states as they strive to strengthen public healthcare systems and ensure access to quality healthcare products. This activity also reflects comstech mandate of promoting scientific cooperation, capacity building, and the sharing of knowledge and expertise among OIC member and observer states. We firmly believe that investing in human resource development and strengthening scientific networks are among the most effective approaches to support sustainable development all across the Muslim world. I would like to express my sincere appreciation to our distinguished resource person for generously sharing their expertise with us today. I thank Professor Dr. Areen Al-Shawed from Jordan for her contribution on quality assurance of liquid and topical dosage forms. We also thank Dr. Tahreem Sara from the Drug Regulatory Authority of Pakistan for sharing her experiences in the quality control of oral solid dosage form. My special appreciation to Professor Dr. Saika Suloma of Burkina Faso for his contribution on regulation and quality control of improved traditional medicine. Ladies and gentlemen, I'd like to acknowledge the efforts of comstech team for coordinating this activity, as well as all participants who have joined us from different countries. Your presence demonstrates the shared commitment of our institution to strengthening pharmaceutical quality standards and advancing healthcare standards across OIC countries. OIC Comstech, the Ministerial Standing Committee for Science and Technology Cooperation, is engaged in many activities all across the Muslim world for improving national regulatory framework and also to enhance capacity of professionals in ensuring the quality of medicines available. I hope that today's discussion will be informative, productive, and will stimulate future collaboration among researchers, regulators, industry professionals, and academic institutions. I wish you all a very successful workshop and look forward to valuable exchanges that will take place during this program. Thank you very much. Assalamu alaikum wa rahmatullahi wa barakatuh.
Thank you very much. We will now proceed with our first technical presentation. Our first speaker is Professor Areen Al-Shawed from Jordan. Professor Areen Al-Shawed is an associate professor at the Department of Pharmaceutics and Pharmaceutical Technology, Faculty of Science and Pharmaceutical Science, the Ashmite University from Jordan. She also served as an advisor for the Technical Committee for Generic Drug Registration and the Post-Approval Changes of Generic Drugs Committee at the Jordan Food and Drug Administration. She has extensive experience in pharmaceutics, pharmaceutical technology, and the regulatory aspects related to medicine quality and registration. Professor Dr. Areen will deliver a presentation entitled Quality Assurance and Liquid and Topical Dosage for Syrups, Suspension, Creams, and Ointments. Dear Professor Areen, please, you have the floor.
Thank you for introducing me in such way. I will share my screen and start my talk immediately. Okay, I think that's okay. Okay. Good morning, good afternoon. Before starting this talk, I would like to express my sincere gratitude for the organizers of this workshop for their kind invitation to talk about this important topic in pharmaceutical industry and drug development, quality assurance and physicochemical analysis of a drug formulation. Here we will talk about the pharmaceutical quality, how and the quality assurance critical rules to ensure the quality, safety, and consistency of drug formulation. Later, we will talk about the most important tools used to analyze the drug formulation. And also, we will talk about how the regulations, such as Good Manufacturing Practice, International Congress of Harmonization Guidelines, support the pharmaceutical quality system. So we will talk about the quality assurance and its relationship to the quality control, good manufacturing practice, and the regulatory framework and guidelines that guide the pharmaceutical quality system. We will talk about also the quality risk management and how the risk-based thinking was integrated into the quality system. We will talk about the physicochemical analysis and the required validation for the process and the equipment qualification. Then, for sure, we will talk about the analysis and the quality control of raw material and finished product, including liquid dosage form, topical, semi-solid, and finally, we will integrate everything into quality assurance workflow from raw material to manufacturing, distribution, and post-marketing surveillance. So quality assurance actually is broader than testing the product. Many people think that quality begins in the laboratory, but in fact, quality begins earlier than this. It's from the thinking about the product. Quality assurance covers every topic that could affect the quality of the finished product with ultimate goal to keep the highest standard of safety, efficacy, and quality of products that are required for their intended use. So it's a life cycle approach rather than single testing activity. It spans from qualification of the supplier, testing of the raw material, including excipients and active pharmaceutical ingredients, to manufacturing, packaging, labeling, storage, and final distribution, in addition to ensure the compliance with the regulatory. So the objectives of the quality assurance is to ensure a product consistency that each batch meets the defined quality standard, to maintain the compliance and adherence to the international and national regulation, to identify and mitigate quality-related risk proactively, and to guarantee product safety and efficacy to protect public health, to promote continuous improvement of the product, and also to ensure that the quality data are complete, consistent, and accurate. One of the main responsibilities of the quality assurance is to write and review and update the standard operational procedures that is required to repeat the activity consistently, regardless who performs this activity. These documents ensure the repeatability and reduce the variability and form the backbone of compliance during inspection and audits. Also, quality assurance rule include the validation process and equipment to confirm that processes and equipment function as intended. We will talk about this later when we will talk about the equipment qualification. The quality assurance team also is responsible for making the internal audit and identifies the gaps and to check if the process is compliant with the good manufacturing practice before the regulatory inspector make their inspection. Also, the quality assurance is responsible for managing documentation and record keeping, because in pharmaceutical industry, if the activity is not documented, it is considered not done. So they must record everything to uphold data integrity and traceability. This includes issuance and review of batch manufacturing record, validation report, logbox, electronic data system, and training files. In addition, one of the most important rules of the quality assurance is to handle the deviation and make the investigation using root cause analysis. And based on this finding, they must define and track the corrective and preventive action to prevent recurrence. The training of the employees is also a role that quality assurance responsible for. The quality assurance oversees the development and implementation of training matrices aligned with job function. They ensure that all personnel receive initial and ongoing training on good manufacturing practice, SOPs, safety, and data integrity. And of course, they record and document everything related to this. Also, quality assurance has the final authority for batch release and disposition. So they review all production and the quality control data to ensure compliance before approving a product release to market. Also, quality assurance manages the change control process by assessing the risk and impact of change in material, equipment, facility, and procedure. They ensure all changes are documented, justified, and implemented in a controlled and traceable manner with appropriate revalidation where necessary. So, the quality assurance is a part of the pharmaceutical quality system that integrates the quality assurance, quality control, and good manufacturing practice using science and risk management to ensure continual improvement and life cycle excellence. The life cycle of the drug starts from the pharmaceutical development, scaling up commercial manufacturing, quality control testing, and quality assurance to reach the market, and the work is continued after the post-marketing to make surveillance for continual improvement. So, generally, the quality assurance build the quality control, check the quality, good manufacturing practice, enable quality, and the pharmaceutical quality system integrate and improve equality. So we cannot talk about the quality assurance without actually talking about the quality control, because they are both work together in a complementary way to ensure the quality of the product. And the quality assurance focus on the process, the quality control focus on the product that analyze. And the quality assurance aim to proactively prevent the defects and errors. The quality control aims to identify, isolate, and define these defects. The responsibility of the quality assurance is everyone working through the life cycle of the drugs, while the quality control, there are dedicated employees and specialized team tasked to detect the defects. So it's like that the quality assurance builds the map and the rules to ensure you don't get lost, while quality control checks the final destination to make sure you actually arrived at the right place. Or we can say that the quality assurance builds the system, while the quality control verifies the output of the system. Again, here we must talk about the good manufacturing practice as a main pillar for the quality assurance, which is a systemic quality management that designed to ensure that products are consistently produced and controlled according to strict quality standard. The good manufacturing practice actually has pillars called the 5B. It focuses on the people, that the personnel must be properly trained and qualified to minimize the human error and contamination. Also, the premises must be designed for logical workflow to prevent cross-contamination and to maintain strict environmental and air control. The processes used must be clearly defined. Every step must be defined, documented in detail, validated to ensure consistent output. The products from raw material to active ingredient and primary packaging should strictly meet the specification, and only approved and tested material enter the production. And also it focuses on procedures that clear written standard operating procedures must be available to be followed by everyone for every operational task, as there is no room for interpretation. Together, the 5 Ps of good manufacturing practice can ensure that safe, effective, and high quality product meet the regulatory standard with reducing cross-contamination by unexpected physical, chemical, or microbial impurities, to prevent incorrect labeling and mixing up labels that could lead to patient or consumers receiving the wrong medicine or dosage, and also prevent the subpotent or superpotent batches where critical ingredients being unexpectedly low or dangerously high in concentration. The pharmaceutical quality system is guided by several global regulation and harmonized guidelines, including Food and Drug Administration, CFR, that regulates the manufacturing, processing, packaging, or holding of drug, the International Congress Harmonization Guidelines, such as Q10 of Pharmaceutical Quality System that describes a comprehensive model for an effective quality management, the European Union good manufacturing practice that covers the good manufacturing practice principles in the European Union, and the World Health Organization guidelines on good manufacturing practice. There are other guidelines, including the ICH Q8 for pharmaceutical development, Q9 for quality risk management, Q2 for analytical validation, Q3A and B for impurities, Q3D for elemental impurities, and M7 for mutagenic impurities. Actually, there are more guidelines must be followed during characterization of the raw material and the finished product. Together, they show the alignment and adherence of the production to the guidelines, which will lead to production of safe and effective drug. The modern equality system is based on the risk management. Okay. On the risk management using different tools such as failure mode and effective analysis that is used to evaluate process systematically to identify potential failure mode, assess associated risk, including severity, probability, and detectability. Also, the diagnosis can be applied by using fishbone diagram or Ishikawa, which is an illustrative root cause analysis tool that maps out and categorizes potential contributing factor, such as material, equipment, method, to a single quality event, and the application of hazard analysis and critical control point, which is a preventive methodology used to identify physical, chemical, or biological hazards, establishing critical limit and continuous monitoring a protocol, finishing with risk assessment matrix that is a structured grid that cross-reference the probability of occurrence against the severity of the harm. And actually, the risk assessment matrix categorize the harm into, or the risk level into low, medium, and high. So the quality begins from the raw material, because the quality of the final product depends heavily on the quality of the starting material. Therefore, the active ingredient, excipients, and solvent must undergo extensive qualification before use. Supplier qualification, incoming testing, and specification compliance are essential components of the raw material quality assurance. So the raw material, including the active pharmaceutical ingredient, must undergo extensive analysis according to the pharmacopoeial monographs or general monographs and international guidelines. The active ingredient must be identified using the official methods such as IR 10-layer chromatography, high-performance liquid chromatography to ensure the molecular alignment with the standard. Also, the properties and characterization of the active ingredient must be analyzed, such as solubility, the physical form, appearance, chirality, melting point, particle size distribution, UV absorption. Everything must be analyzed. In addition to the assay to determine the exact amount of a drug and if it meets the specification written in the official monographs or not. Also, the impurities must be defined and sometimes qualified, including the synthesis-related impurities and degradation product, the inorganic impurities such as heavy metals or any other inorganic contaminant remaining from manufacturing. Also, the residual solvent to ensure that volatile organic chemical used during synthesis are within the safety limits according to the guideline Q3C. And nowadays, there is a huge focus on identifying and reporting, qualifying also the genotoxic impurities according to the ICH-M7 guideline, and also the nitrose amine impurities because of their potential carcinogenicity. Of course, and also the microbial limits of the active pharmaceutical ingredient must be identified to check the total aerobic microbial count and the yeast or mold microbial count. Stability also must be performed, including long-term stability at 25 centigrade and 60% relative humidity, or 30 centigrade and 70% relative humidity, depending on the zone, in addition to accelerated stability at 40 centigrade and 75% relative humidity condition. In the stability studies, everything, including assay, impurities, microbial bioburden, must be checked to ensure their consistency and adherence to the specification. Raw material, including an excipient, must also be qualified. We know that the excipients don't have activity, but they affect the quality of the finished product. They may affect the solubility, dissolution, the stability, or the manufacturability of the drug. So again, here, they must be tested for their appearance to verify their physical state, color, crystalline or powder structure, amorphous structure. Also, the solubility and behavior in different solvents such as water and alcohol to check their solubility. There must be critical analysis for their density, optical rotation, parameter, viscosity to verify the biological behavior, especially if they are used for suspension. Also, again, here we will focus that many tests are general, must be made for drug substance, excipients, and finished products, such as the identity. Also, the moisture and volatiles must be checked in the excipients using Karl Fischer for water content or loss on drying. The excipients must undergo analysis for their pH and buffering capacity for determination of their ash and, of course, stability, as we said before, and presence of heavy metals, particle size distribution, especially if they will be used in suspension. Also for microbial bioburden and the absence of objectionable microbial species such as Staph aureus and Pseudomonas aeruginosa. The tools, most commonly tools used to analyze the drug substance and finished product also include the spectroscopic technique, such as UV visible spectrometry, and FTIR, Raman, that measure the light scattering and fingerprint for material identification, quantitative assay, and excipients compatibility, chromatographic method. These methods are actually, they are official, mentioned in the monographs, must be used to identify to measure, the assay and impurities in the drug substance and finished product, such as HPLC, ultra performance LC, and gas chromatography, which is used mainly for volatile gas analysis. The thermal analysis, including differential scanning calorimetry to check the phase transition and heat flow, the thermogravimetric analysis that measures the mass change versus temperature. These methods used for polymorphism study determination of the melting point and physical compatibility. Particle size characterization is important, especially for suspension. Laser diffraction, optical and electron microscopy can be used to check the particle size and study the bioavailability of different particle size, the physical dispersion structure, and also these instruments are used to check the zeta potential, which gives indication about the stability of the suspension and nano-suspension. Of course, radiological evaluation must be performed using rotational and capillary radiometry to check the viscosity profile, the type of flow, and the formulation yield stress. The methods used cannot be trusted if it's not validated. So the guidelines, ICH Q2 guideline, guide the analysis, the validation of the analytical method, and mention the parameter that must be checked, including that the method must be specific to the analyte in the presence of impurities or matrix component, that the method must be accurate. So the accuracy or closeness of agreement between the value and the values founded by the procedure must be checked, in addition to precision, which measures the degree of scatter among individual test results, including repeatability and intermediate precision. Actually, again, the method must be linear which means there is ability to obtain test result directly proportional to the analyte concentration in the sample. The range also must be defined, which is the interval between the upper and lower concentration with suitable precision, accuracy, and linearity. The robustness must be reported that show the capacity of the method to remain unaffected by small deliberate variation in method parameter. And also there are two important parameters, the limit of detection that measures the lowest amount of analyte detectable in the sample, but not necessary to be quantified, and the limit of quantification, the lowest amount of the analyte can be quantified with suitable accuracy and precision. The equipment also must be quantified to make trust that the results we are taking are reliable or not. This includes, as the USB chapter 1058 guide, that the design must be qualified to verify and document that the proposed design of the instrument is suitable for its intended application. Installation qualification also to ensure that the instrument is received as specified and it's properly unpacked, installed, and configured in a suitable environment facility. The operational qualification verifies that the analytical instrument functions correctly and perform as expected across its entire intended operational and environmental range, and the performance qualification to confirm that the instrument consistently and reproducibly performs in accordance with specification appropriate for routine daily analysis over time. So if the process process validated and the equipment qualified, we will have the trust with the analytical results. So as we said,
3 minutes. 3 minutes.
Okay. Okay. So as we said, all the active pharmaceutical ingredient, excipients, and the finished product must be specified to check if they meet the specification or not. For syrups, there is a specific characterization must be performed, including the appearance, the odor profiling, the fill volume verification to control the minimal volume requirement for multi-dose packaging, and also to check the clarity, the sugar content, Brix test, 1 degree Brix test, and specific gravity. These two tests give indication about the amount of sucrose used in the syrup. Of course, as we said, there are general tests must be applied, including the assay, impurities, preservative efficacy, the pH stability. The delivery must be checked that mass deviation limits from multi-dose container must be defined in addition to microbial bioburden, target pathogen screening, in addition to stability, as we said, which is universal test for everything. For suspension, there are parameters must we focus on, such as sedimentation volume, particle size control, to check the homogeneity of distribution and to prevent caking, in addition to pH stability ranges. For chemical control and assay, it's universal to check the identity assay, impurity, degradant, stability, microbial count. Also in suspension, we focus on also in dissolution, the release characteristics of the drug, because it's highly affected by the particle size. For finished products of semi-solids, such as ointments and creams, the physical characteristics is very important. to evaluate the viscosity and physical separation or gritness. Also, the spreadability, which measure how easy to spread the product on the skin, and extrusibility to measure how it's easy to remove the product from the container. The rheological behavior is very important to check the viscosity, thixotropy, and flow. Of course, the base suitability must be checked to ensure the compliance of the vehicle base with the topical semi-solid specification. Also, in vitro release testing and in vitro permeation testings are required when appropriate, in addition to universal tests that we talked before about syrups and about suspension. So the quality assurance workflow, as we said, is not a single phase. It's a life cycle, starting from qualification of the raw material and the including excipients and active ingredient and solvent, qualification of the supplier, if they are trusted enough to get the product from them or not, to check the incoming material if they meet the specification, general and specific specification mentioned in the guidelines. Also, to apply the good manufacturing practice control, to check the formulation process, such as mixing, to check the mixing time, agitation efficiency, temperature during mixing. Also, there's something important to do. It's the in-process quality control, depending on the formulation. For syrup, we have to check the pH, viscosity, clarity, color consistency, specific gravity. For suspension, particle size, sedimentation, uniformity, pH, and density are important parameters to be checked during formulation. For ointments, the temperature, homogenization control, appearance, examination, viscosity, homogeneity, particle size, density, and also deaeration check are important for the checking the quality of the product. The process continue with the filling and packaging to check the fill volume, the container closure integrity, and the label control. The finished product must be tested, as we mentioned. Okay, and the stability studies must be performed. And after that, the quality assurance team review everything related to the product and make the decisions about batch release. They do the batch record review and check the out of trend and out of specification and investigate if any happen. Also, this continue with post-marketing monitoring to ensure the continual improvement of the product. So to conclude, quality assurance is a life cycle approach that ensures pharmaceutical products consistently meet safety, efficacy, and quality requirements. Physicochemical characterization of the active pharmaceutical ingredient, excipients, and finished product is essential for product development and quality control. The analytical method must be validated. Also, the instruments must be qualified according to relevant guidelines to ensure reliable and accurate results. The quality risk management supports proactive identification and control of potential quality issues. In-process controls, finished product testing, and stability studies are critical components of a robust pharmaceutical quality system, and effective quality assurance integrates good manufacturing practice, compliance, scientific understanding, and continuous improvement throughout the product life cycle. There's a saying in pharmaceutical product that quality cannot be tested. It must be built and integrated from the beginning. That's it. Thank you for your listening. Your question, of course, feedback, inquiries are welcome. And if you need any elaboration of any covered topic, also you are welcome to contact me directly. Thank you.
Okay. Thank you very much, Professor Irene, for your insightful presentation and for sharing your expertise with us. quite long but very interesting. So I don't know if my colleague have the question, Dr. Farah, Dr. Abdul Rashid, if you have a question.
Dr. Arin,
Yeah, of
very
course.
good. Very good presentation. My name is Dr. Abdul Rashid. By profession, I'm a pharmacist and working as a consultant health initiative in OIC Comstech.
Thank you.
I have two questions. One question have two parts. The first question is, how many pharmacy education are available in Jordan? This is one part of the first question. How many pharmacy education departments are available in Jordan? And the second question, the second part is that how many pharma industry they are licensed by Jordan FDA? This is first question. My second question is that I am a former president of Pharmacy Council of Pakistan. By the platform of OIC Comstech, we have conducted 3 meetings. That OIC $15 platform means among 57 Muslim countries, 15 Muslim countries are in such a condition that have very potential to do work in science, technology, and different areas like pharmacy, engineering, medical. So among those 15 countries, all Arab countries are included: Pakistan, Iran, Turkey, Azerbaijan, Jordan, including Jordan, Indonesia, Malaysia, and other countries. We have conducted 3 meetings that OIC $15 platform, that is 15 Muslim countries, they should have, they should make a design, a framework in which 15 Muslim countries, they should have the same syllabus of pharmacy education. We have already conducted 3 meetings. I request you, my advisor, Professor Mudassar Asta, she's also a university professor and acting vice chancellor. We want to continue this activity that 15 Muslim countries, they should have unique curriculum for pharmacy education under the umbrella of OIC $15 platform. Would you available for that activity? Number one.
Yeah,
As expert in pharmacy education, can you give the time either virtually or physically? That if we
next
make
meeting
it,
is on 25th.
yes,
Yes, 25th of this month. Next
Good.
meeting.
next meeting is on 25th of this month. So I will request you that you either you will be available either virtually or physically for this activity to design a framework for a common curriculum of 15 Muslim countries. So this is the second question.
Thank you for your questions. Of course, the pharmacy is the same every way and should be covered by the same framework. But actually, Doctor, here in Jordan, we are not agree on the same curriculum. By the way, different universities have different curriculum depending on their, let's say, trend for their students. For example, in the Hashemite University, we depend on tracks at the students' 5th year, if they will go for industrial part or for the management. That's why we have different curriculum for different students at the same faculty here in Jordan. But, you know, we are open for collaboration in Jordan, here in Jordan. Thank you. I don't know if I answered your question or not.
No, you are very well. You answered the question. Thank you very much.
Thank you very much, Dr. Farah. I don't know if you have questions. Okay, thank you.
No, I will ask the question.
Yes, I have a few questions online.
Okay, let me ask.
Okay.
My question is that you are testing everything in vitro. Are these things similar in vivo?
Sorry, pardon me, please. We are talking about the testing in vitro and in vivo?
Yeah, in vivo the things are different. So how do you judge that the things in vitro are similar in vivo?
Of course, the applicant for the drug product must go for their test, not just in vitro. They have to go for human studies, clinical trials to check. As a part of the advisory for the generic drug, we have to check the bioavailability of the drug and the comparative studies with the originator drug, if they will pass or not, gives the same bioavailability or not.
Thank you.
Okay, thank you, Professor Arin. I
add one thing, that for the session of the medicine, there is no need of conduct of the in vivo studies. During clinical trials, we conduct the in vitro and in vivo studies. For when we come for the purpose of the session, there is only in vitro study, in vitro data and in vivo data submitted.
That's why.
Thank you very much. I have two questions from our online participants. The first question is, considering the challenge faced by many pharmaceutical manufacturers in maintaining consistent product quality, what are the most critical quality control parameters that should be prioritized for liquid and topical dosage form?
As we say, they must meet the specification. There is no thing that they meet some specification and not other specifications, okay?
Yeah.
The specifications are already guided by the monographs. If there is a monograph for the products, for example, we use the USP monograph or the European monograph or the general guidelines. For me, if I want to check the profile of a product or the documents of a product, I will check the assay, degradants, including, as we said, nitrosamine and genotoxic impurities, if they align or not. In addition to their stability, if they keep stable over the time or the shelf life of the product or not.
Okay, thank you. Second question is, from your experience with pharmaceutical regulation and generic drug evaluation, what are the common quality issues observed in liquid and topical formulation, and how can manufacturers and regulatory authority work together to address them?
If there's defects, you say, I don't get it exactly.
I say, what are the common quality issues observed in liquid and topical formulation, and how can regulatory and authority work together to address this common issue observed?
Actually, the regulatory is very strict about the product specification. We don't have the room to give opportunity to not meet or not adherence to the specification used. For topical, maybe we see microbial growth is a problem for us.
Okay.
Also, the stability is a big concern for semi-topical, yes.
Okay, thank you very much for your insight and for your presentation. Look forward to meet you again. Okay, we will now move to our second technical presentation. Our second speaker is Dr. Terim Sara from Pakistan. Dr. Terim Sara is the Additional Director of Quality Assurance and Laboratory Testing at the Drug Regulatory Authority of Pakistan. Through her role at DRAP, she contributes to pharmaceutical quality assurance, laboratory testing, and regulatory oversight area that are essential for ensuring the safety, quality, and effectiveness of medicine. Dr. Terim will deliver a presentation on quality control of oral solid dosage form, tablets, and capsule. Dear Dr. Terim, you have the floor.
Esteemed representatives of regulatory authorities, respected colleagues from academia and pharmaceutical industries, ladies and gentlemen, Assalamu alaikum and a very good afternoon. My name is Tareem Saya. I'm currently working as National Regulatory Agency, which is called Drug Regulatory Authority of Pakistan. I have total 25-year experience in dossier evaluation for the marketing authorization and currently working in quality assurance and lab testing. My today's topic is quality control test for the oral solid dosage form. Main emphasis is on tablets and capsules. As we all know that oral dosage forms, particularly tablets and capsules, remains the most widely manufactured and utilized pharmaceuticals product worldwide. Their quality must be consistently demonstrated through scientifically sound, pharmacopoeially compliant quality control testing. So my main topic is on the quality control, not on the quality assurance. So my main emphasis is on testing, basically on the testing. So quality control is combination of all these three parameters, which is very well explained by Dr. Areen. It's the combination of CGMP. CGMP is current good manufacturing, starting from documentation and data integrity. Then it comes materials and analytical testing, and then equipment and method validation. All these three parameters combines make a quality control department. So quality control, as we all know, is not an act, it is an habit. Quality control makes it measurable. Any quality which is measured through test, through sampling, through specification, that is called quality control parameters of that particular dosage form. So general definition of quality control is a part of good manufacturing which is concerned with sampling, specifications, and testing. It ensures that necessary and relevant tests are actually carried out and that materials are not released for use nor product released for sale or supply until their quality has been judged to be satisfactory. Core objective starts from the integrity of the raw material, which has also been explained very well by Dr. Aline in the case of syrups and dosage form. Here, quality control majorly is powder, not limited to, ensures that incoming material meets identity, purity, and quality standards. Precision in the manufacturing process to cater the process generated impurities, verification of finished product specification, which is the official and non-official test, safety and efficacy for the patient. All these three core objectives leads to the main objective, which is safety and efficacy of that particular dosage form. So quality control is a gateway between the manufacturing and patient, starting from the tests, then evaluations, then decision, then release, and ultimately quality reaches to the patient. Why QC is necessary? Again, when we are talking about the medicines, its mainly concern is patient and ailing humanity. So the patient safety and therapeutic efficacy. So patient safety prevents substandard and toxic product to reach to the market, and efficacy ensures the drug delivers for the intended dose at the right rate. The third one is the legal compliance, adherence to the specification, either it is from the part of National Regulatory Agency or through the specification which has been catered by the pharmacopoeial standards. Brand integrity, this is at the part of the firm. If a product is of quality, that may build the public trust in the industry. Now, types of oral solid dosage form, when we discuss about tablets and capsules, so oral solid dosage form ranges from conventional tablet and capsule to advanced modified release formulations. Different oral solid dosage form possesses distinct physical and release characteristics. Quality control requirement must be tailored to specific dosage form architecture. Here is some examples. In tablet, the representative form conventional, which is compressed tablets, simple, film coated, again have some polymers coated on that, chewable that can be chew, and the other one, the more specific and the complex one is immediate and modified release. Capsules starting from hard gelatin to soft gelatin, rapid or delayed release, systemic delivery. Other solids is powder granules and lozenges, local or systemic absorption. Now, the complexity of and variety of the oral solid dosage form, as we discuss about tablet and capsule, is start from the conventional tablets at first with immediate release. Then it comes to the tablet coated with a thin polymer film to improve appearance and stability. Again, another coated tablet that is enteric coated to protect this degradation by the stomach acid and release in the intestine to get the benefit of efficacy. Third one is coated and sugar coated tablet to improve taste and easier for swelling for bitter taste drugs. Then is chewable tablets. These are mainly used for kids. Disintegrates rapidly, direct compression that disintegrate rapidly in mouth. Narrow therapeutic index drugs and the drug which is mainly used and most famous is Angisid. And other one is placed under the tongue for rapid absorption, then placed in the cheek pouch for absorption. The next one is designed for extended or delayed drug release, dispersible tablet in water before administration. So all these 6, 7, 8 parameters shows the diversity in tablet dosage forms. The next one is capsules. In capsules, we got two pieces hard shell capsules, then soft gel capsules, then designed to release the drug in the intestine again. It contains pellet, then multiple particulate for oral use, then spherical multiparticulate system for controlled release. Each oral solid dosage form has a unique design as we talk about conventional to modified release and release profile. Therefore, quality control testing must be specifically adapted to ensure a consistent performance of safety and therapeutic effectiveness. Sequence of quality control testing, as Areen has explained, because quality assurance starts from the raw material. Similar is the case, quality control starts from the raw material. A very good and pure raw material leads to a very good finished product, provided that the manufacturing degraded product shall be catered. So raw material testing is very important as well as with the excipient, which has been elaborated in my previous presentations. Raw material testing, verification of API excipients before production. This has been discussed in the previous presentation. In-process quality control, monitoring during manufacturing to control process, finished product quality control, and then stability monitoring. So the process flow quality control starts from the raw material, then from the manufacturing, leading to IPQC, which will affect the quality of finished product and then its release. So IPQC, in-process quality control, and finished product quality control are highlighted as critical control points in the manufacturing process. Each stage required documented evidence and compliance before proceeding to the next step. These documented evidence is the GMP. Each and every step should be documented and should be verified. So stages of quality control test, as I have explained, it is in-process quality control that affect the batch release at the end of finished product. So in-process quality controls involve testing performed during the manufacturing process. These processes are usually called as non-pharmacopoeial test or conventional test. It leads to continuous process monitoring, real-time intervention capabilities conducted on production floor. So this in-process quality control doesn't have to go to QC lab. Primary goal is to monitor critical parameter and adjust the process in the real time to prevent batch deviations. Batch release tests are usually the pharmacopoeia test, which is mandatory, that contains and that relies mainly on the in-process quality controls. So conducted after complete production and packaging, conformity of final validation, regulatory compliance, that is to generate the certificate of analysis of final product based on the specification approved in the pharmacopoeia as well as by the NRA. Serve as the final gatekeeping steps to verify that batch meets all the regulatory and compendial requirements. This mainly includes stable batch stability studies. So IPQC test for the tablets and capsules. Keeping in mind the time duration, I have not been elaborated these tests. I only mentioned this test has already been mentioned in the pharmacopoeias and also clarified in the oral solids form explained by Dr. Areen. So in the case of tablets, it is physical appearance, which under the heading of physical appearance, color uniformity. As I have shown in the diagram, a tablet, a dosage form should be appealing. Since it is a tablet, So when you open a candy, it should be appealing. So medicine should be appealing for the patient. Shape consistency, odor assessment, identification mark, which is also called logo, that should be in each and every tablet, it should be uniform. Surface defects, coating integrity. The next one is granule evaluation. Prior to come into the final dosage form as a tablet comes from powder, then to granules, then to compress, and then in the form of tablet or capsules, whatever the dosage form is. So granule evaluation at this stage is the main importance is moisture content, which affects the assay of finished product. Particle size distribution, which affects the release pattern. So each and every in-process quality control test, which is called conventional test, will affect the final companion test. Next one is flow properties. This again will affect the content uniformity as well as the release characteristic inside the body. Bulk density, compatibility, compressibility index, and angle of repose. The next parameters is weight variations, dimensions, hardness, friability, and disintegration time. Now we finally move into today's topic, classification of quality control test, physical and chemical. The physical test includes hardness, friability, disintegration, weight variation, thickness, dimension, and appearance. All these tests are physical tests, but all these tests have the effect on the finished product quality control. So chemical test, which basically occurs in the QC lab, these are evaluate chemical identity, strength, purity, and performance dosage form. Here I have to mention that chemical tests are usually compendial tests that is mandatory to perform, and all the product quality and safety depends upon it. So both these tests will come and ensure the quality, safety, and efficacy and consistency of pharmaceutical product. Now, physical test ensures tablet capsule is made correctly, appealing, right dose, no edges break, nothing. The chemical test ensures product as what it should be. That is, the product is made for the effect of safety and efficacy. Together, they ensure safe, effective, and high quality medicines. Now, this is the categories which has been defined in the pharmacopoeias. Again, I am here came up with the official pharmacopoeial test. As I explained, these are the chemical tests. Test mandated by the official compendia. Application establish identity, strength, purity, quality, and performance of a dosage form. Examples of these tests mainly but not limited to identification, assay, related substances. These are impurities, dissolution. Dissolution explained as release pattern of the drug inside the body. Disintegration, uniformity of the dosage form, that each tablet contain equal amount of intended dosage form. Water content. Unofficial and non-compendial test, internal standards used by the manufacturer to monitor process consistency. You can also say that these are the formulation development test. Establish additional physical, mechanical, formulation, and process-related characteristics that support product understanding and process control. In these tests include hardness, thickness, diameter, loss on drying, particle size distribution, and density. Here again, particle size distribution has the effect on the dissolution. Hardness has the effect on the chemical test of disintegration. Loss on drying has the effect on assay. Density has the effect of dissolution as well as uniformity of dosage content. Now the official chapters, which you can get the detail of each and every test along with its equipment and its requirement. Dissolution is mentioned in the journal chapter 711 and of the USP Pharmacopoeia and Appendix 12B of the BP. This is mainly but not limited to, typically Q is greater than or equal to 75% or 80% within 30 to 45 minutes. I have only given the example of immediate release tablet. Immediate release. Uniformity of the dosage content, it's included in the chapter USP chapter 905 and BP chapter 12C. Average weight plus minus 5% to plus minus 10%. Yes. Back. Oh, back.
Back down, loosely.
Back.
Check this.
Back. Go back, yes.
Yes, more back. Okay.
नहीं अभी और भी जाना है बैक more और मजीद yes
Mazid.
बस
Drug essay specific monograph. Specific that is mentioned on the monograph for each and every chemical. Usually 90 to 110% level claim in USP, and BP has a strict limit of 95% to 105%. Disintegration is mentioned. It's mentioned in the slide. You can go and check it. Acha ji. To quality control test for the tablets. Again, it's identification of the raw material which has been done through HPLC, UV spectroscopy, IR or TLC. It is mentioned on the individual monograph of the product as drug substance. Assay is related to the drug product. It's usually performed by HPLC, UV, titration, and typical acceptance range, which is exemplary given as 95 to 105%. Then is critical parameter is impurity. Impurity is related to the safety of the product because drug substance related impurity is genotoxic. Ensure product safety and stability limits are specified in USP and BP monograph of for the impurities. Volatile content, measure the residual moisture of solvent, loss on drying, and Karl-Fischer titration is the apparatus important for stability and shelf life. Now I have mentioned each test, how to perform each test. Weight variation test, weigh 20 tablets individually, calculate average weight, which is mentioned as AW, compare the tablet, get the calculation, and when the acceptance limit is the tablet which is less than or equal to 80 milligram, weight variation of 20 tablets should not be more than or less than 10%. Rest 80 to 125 milligram. Plus minus 7.5%, and 250 milligram it's plus minus 5%. If weight variation fails, content uniform test must be performed. Disintegration test is conducted to measure the time required for a tablet to break apart into particulate under standardized fluid condition. Temperature is test conditions are 37 plus minus 2 degrees centigrade, that is the body temperature. Sample size 6 tablet, and medium is simulated gastric fluid. Acceptance limit for the uncoated tablet is 15 minutes, film coated is 30 minutes, and for enteric coated is 60 minutes. Now the most and the critical test is dissolution test, because this test affects the product release pattern inside the body. How the drug is available in the body to perform its intended purpose. So these are the apparatuses, 4 type of apparatus. The most common is basket and pedal type. What we do is it's categorized into 3 stages. S1, we take 6 tablets. Then each unit should be or no is equivalent to or not greater than Q plus minus 5%. Q is the value of the drug that has to release, and it is expressed in percentage. Here it's not plus minus, it's only plus 5%. If test fail at this stage, then you go to stage 2, where you get 6 tablet plus 6 tablet, and then average 12 unit is not greater. Q, first of all, we have to check the Q. Q should not be equal to minus 15%. Then again, if at this stage the product fails to, then we have to conduct the double the quantity, 12 plus 12 tablet, and average of 24 unit, not more than 2 units goes to 15%, and none of the unit goes above to 25%. Now the acceptance criteria, where Q specified amount of drug dissolved in monograph expressed as percentage of label amount. If dissolution passes, results meets S1, S2 and S3 criteria, fails if result do not comply to the above test. The details of these tests is mentioned in USP Journal chapter 711 under the heading of dissolution and British phonograph dissolution test. Again, hardness and friability, drug assay is mentioned in 621 Journal chapter HPLC and spectrophotometric in USP visible. The acceptance limit and equipment is mentioned here. For BP standard for modern monograph provides qualitative data, quantitative data through peak area, area under the curve, and for USP its absorbance, Beer-Lambert law. Quality control test for capsules. Capsules also is appearance, shape, size and shape, identification market, fill weight, and content uniformity. API distribution check. Finished product assay, dissolution, disintegration, and stability. Content uniformity test of the capsule is mentioned here. We have to take 10 capsule, check the individual weight. The criteria is mentioned, not more than one of the individual value outside the limit of 85 to 115% of average value and none is outside the limit. Here you see the area with the red color tells us about the failure of this test. 20 capsules, 3 goes out of the range of 85 and 1 goes out of the range of 115. Capsules disintegration and dissolution, again hard gelatin capsule 30 minutes, international pharmacopoeia limit and BP limit is 30 minutes. Soft gelatin capsule 30 to 60 minutes and direct coated 60 minutes as in the case of tablet. Apparatus, basket, pedal, same S1, S2 acceptance criteria as mentioned in the tablet. Comparison of BP USP more than both have same test that is mandatory for the tablets and capsule. Difference between pharmacopoeias are minor. The objective remains same across all standard quality, safety and efficacy. Future trend in QC 2026 evolution. Now we move toward where traditional QC approach inside the lab changes to future QC landscape that PAT, continuous real-time monitoring, quality by testing, replaced by quality by design. Manual or destructive testing will be replaced by AI-driven analytics and non-destructive sensors. Regulatory static pharmacopoeia limits dynamic control within the defined design scopes. Key home messages or conclusion is IPQC prevents defect during manufacturing and finished product confirms quality before release. Tablet and capsules must comply pharmacopoeia standards. Quality control is essential for ensuring safe and effective medicine. So quality cannot be tested into a product, it must be built into it. As I mentioned earlier, it's not the act, it's the habit. Quality is not the act, it's the habit. Thank you very much.
Thank you, Dr. Terim, for your beautiful presentation.
Pleasure.
Also, thank you for joining us today. Thank you very much. We are very pleased to have you with us.
Thank you.
If there is any question,
Online questions.
online questions, I have 2 questions from online participants. The first question is for oral solid dosage forms such as tablets and capsule, what are the key quality control tests that should be routinely performed to ensure product quality, efficacy, and compliance with pharmacopoeia standards?
The pharmacopoeia for tablet and capsule varies from each monograph and it's very much mentioned in under the individual chemical for drug substance as well as for the finished products.
Okay, thank you. For the second question, from a regulatory perspective, what are the main challenges that quality control laboratory face in ensuring the consistent quality of oral solid dosage form, and how can this challenge be addressed?
What we got through being in the National Regulatory Agency, the majority of the product comes in the substandard quality on the release pattern as well as on the assay. The main issue is stability studies. If a product is not stable, it will have these issues.
Okay, thank you very much for your presentation, Dr. Abdul Rashid.
It is an art. I am Dr. Abdul Rashid. I have 42-year experience in pharmacy profession. I worked as a teacher in a university, and also I'm a retired director of Dr. Authority Pakistan, and also the ex-president Pharmacy Council Pakistan. It is a hard question, but it is a comment. I think I am also a student of quality control and quality assurance. This presentation is a very comprehensive, and the regulators of not Pakistan but other regulators of the Muslim countries, they can take benefit from this presentation. Dr. Tarim Sara, thank you very much.
Thank you. Most welcome. Thank
Okay.
you.
Thank you very much.
Opportunity.
Thank you very much, Dr. Tarim, for joining us. Thank you very much. We will now move to our third and last technical presentation. Our third speaker is Dr. Isiaka Sulama from Burkina Faso. Dr. Isiaka Sulama is the Director General of National Pharmaceutical Regulatory Agency of Burkina Faso. He is also a researcher at the Institute for Research and Health Sciences and at the National Center for Malaria Research and Training in Burkina Faso. His expertise combines pharmaceutical regulation, research, and public health with particular relevance to improve medicine quality system. Dr. Sulama will present on regulation and quality control of improved traditional medicine. Dear Dr. Sulama, thank you for joining us. We look forward to your presentation. You have the floor. Thank you, Dr. Sulama. Dr. Sulama is online.
Yeah, thank you very much. Can you hear me?
Yeah, we can hear you. Go ahead.
Okay, thank you very much. Thank you for really giving me the opportunity to be part of this online training on quality assurance and physicochemical analysis of drug formulation. And I will be presenting on regulation on quality control, specifically focused on traditional medicine. With let's say focus sometime on what is ongoing in our country. Yeah. Thank you very much. So I will try to be to really work on this plan, key definition, also making a very brief introduction on the traditional medicine. I will be focusing most of my talk on the quality control of improved traditional medicine, but also looking how the regulatory framework is organized around the traditional medicine. And I will be handing my presentation with a very key take-away message. So just a very brief definition on two most important words. African traditional pharmacopoeia is really defined as the body of knowledge, practice, techniques, preparation and use of plant, animal, or mineral substance that serve to diagnose, prevent, or eliminate physical, mental, or social imbalance. They constitute an African therapeutic heritage. This is very important to really keep in mind. So in terms of definition for the traditional medicine, this is any medicine developed by a traditional health practitioner or researcher using knowledge or information derived from the traditional pharmacopoeia. The medicine, in terms of real organization, are divided in 4 categories. We see them in the next slide from category 1 to category 4. So let's say historically, very difficulty on how to really guarantee the quality and repetitivity of herbal care long drove making the medicine, traditional medicine, really a second round when you compare to the conventional medicine. So, however, today when you look at there is a many technology make it possible to really ensure the quality from raw material to finished drug now. And in terms of really traditional medicine, what is very important is the best and the excellent quality of the plants that were being used for the traditional medicine. And this will be based on the growth condition of those plants and the property dry or those plants, but also how those plants are stored and used before the expiration of date. And a poor quality plant will represent a waste of time, but also money. But above all a dangerous situation for those will be using those plants. So it's very important that the production of plant-based medicine follow established standard, but also a validated method in order to be sure that we are guarantee the pharmaceutical quality and the productivity. So importantly, quality control will check the raw material and the finishing product based on the minimum requirement. This is very important because on top of everything, we need to have a minimum requirement that the user should fulfill to be sure at least we are guaranteeing the quality and the productivity of medicine. So in terms of improved traditional medicine, of course, we know that this is one of the areas now growing in the continent. We know there is some problematic issue around that in terms of quality control, but also in terms of regulatory aspect. So we will really look at now in terms of quality control, but in the regulatory framework, what is now ongoing, what is now being done by the different countries to improve, to really take now ahead using traditional medicine. So what are the improved traditional medicine? We know that in Africa, most of the population between 70 and 80 of Africans are really rely on traditional medicine as a primary health care. This represents the bridge between ethnopharmacology and the formal pharmaceutical product that we have now. So in terms of definition, we can say the traditional, the improved traditional medicine is a traditional remedies proceed into standardized dosage form, but also to improve quality, safety, and efficacy while retaining the ethnomedical basis. So there are three important steps when you really want to move from traditional medicine to the improved one. The first step is really the origin of the plant that are being used, deriving from documented traditional healing practice. And the second step is to standardize the formulation, to get a fixed dosage, to define and extract concentration. And the third very important step remain the validation, how really to look at during the preclinical step to physicochemical profiling, but also the toxicity screening, because at the end we have to be sure that the product we are now putting to market is not toxic for the user. So we'll be looking to those 6 boxes, very important. Quality control, that is the key. We know that any failure in quality control will really affect the health of those users. But we also be looking to one of the most important issue in traditional medicine also is the contamination and the adulteration. And we look into the regulatory landscape around the traditional medicine, but also a focus on the clinical evidence, what is being done there, and all insuffisance cases that we can really highlight. Most important also is the pharmacovigilance gaps to looking also during this presentation. And we'll also discuss, sometimes there are undeclared pharmaceutical product part or include in the improved traditional medicine. And this is very important. Sometimes it's not known, that can really affect as well the health of the user. So what are the objectives of the quality control for the improved traditional medicine? The objective is clear, is to guarantee the safety, the efficacy, and the reproducibility of the herbal product. Based on looking to the botanical identity of the plant that are being used, you have to certify the botanical identity of the plant, but also the purity of a plant that are being used. We need to really be sure that we are detecting adulteration, substitution, or contaminant as a very first important step when you are looking to the quality control. And definitely, we need also to look at the third position, the active ingredient content. We need to assess the content of an ingredient of a chemical marker to be sure at least we know exactly what are the active ingredients in your improved or traditional medicine. So we know that there is some missing when we come to quality control regarding the improved traditional medicine. Of course, we know that the issue related to the species authentication. We also know that in terms of profiling chemical and active component identification, there is sometimes some missing information there. We also know that in terms of quality control, we need to face to some contaminants like heavy metal, but also pesticide and aflatoxin that can be also part of the product we are putting on the market. We also have to looking on the microbial safety testing, look at to the bacterial, to fungal load that are really linked to the traditional medicine that we are using, and look at to the stability and the dose for some standardizing studies. The most important thing is when you are dealing with that, we have also to consider the WHO framework. The good agriculture and collection practice should be also learned and to be part of the development of improved traditional medicine. The GMP is also key. That really those really work on this area have to be considered when we are really trying to improve the traditional medicine to put it in the market. Because at the end, the consequence of really not considering missing some of important key point on the quality control is the variable doses between batch, for instance, unpredictable efficacy, but also the inability to obtain market authorization under a national framework.
Please, Dr. Sulama. Please, Dr. Sulema. Okay. Outline. Dr. Sulema have network issue. You can wait for a few minutes. He will. Hopefully, he will be back. It's okay.
Welcome.
Go quickly.
Right, yeah.
Yes, I'm sorry. Yeah. Okay, so really now I was really discussing the characteristic of a botanical powder, where I was saying that it's very important to really go for that by a different step. The first is really looking to the color, the odor, the taste, and based on a microscopic and organoleptic examination. But also using the microscopic observation, you can really assess the powder fragmenting from a plant, where you can look into some of the content of a plant, the parenchyma cells, but also the stoma. And most important also is to consider to look at to the size of a particle. And this is the measurement that can be done. And there is 3 different sites that really characterize the powder when you are really tackling the issue of a medicine product. The size should be can be under 250 micrometer or between 250 and 500 micrometer or more than that. This is very important to characterize the size of a particle being a part of your medicinal product. So we also need control quality control issue to look at the physical-chemical parameter, meaning that water contents, the total H, but also the pH of your product. This is very important because there is a expecting result that you need to really be in between to be sure at least you are guarantee the quality of your product. But also look at some of to screen what we call the physicochemical parameter is also very important to look at them. They are essential when we are really tackling the medicine because the traditional medicine are different components. So we have to look at the polyphenol, to the tannin, to the saponin, to the glycoside, to be sure that at least you can test them and you know exactly what are the proportion. And the different tests are really very different tests according to the type of your chemical group. For instance, when you look at the polyphenol, we need to really look at the color. It should be a blue coloration as opposed to one, showing that you are in your medicine, your plant, a polyphenol. So we have to look at those screening when you are really dealing with the quality control. This is the same when you look at some of our components as well, because the first one, for instance, is a blue component. But when you look at some of the physicochemical group, for instance, the positivity of the presence, meaning that the presence of those components in general are characterized by also the color, but in general, for the sterol, for instance, is red-brown coloration that we can see when we are really tackling them. Also, the triterpene, you will see a red color, for instance. So the sugar also should be part of your quality control to be sure at least they are there and what are the proportion of that content in your traditional medicine plant. And in terms of method technique that can be used, there is a thin layer chromatography that can be also used as a technique to look at, to make your plant fish pink. So the chromatography of the preferred technique that can be used. For instance, you can also read your chronogram. For instance, look into the figure at your right, you can see exactly how we can really look at the plant fingerprint using the TLC technique. You can look at the number of the cycles that you have in your profile, but also look into the position of those, but also the color that you are seeing when you are really looking to the characteristic of your plant. And this is very important when you are really dealing with the quality control. You need also to assess your active ingredients that are part of your product. This is very important. A very different technique that can be used. You can use the HPLC, the high performance liquid chromatography, but we can also use the TLC that we described that in the previous slide. It depends on really the level of quality and you want to reach. By using, for instance, the TLC, this is an innovative method that covers the electronical image and the chromatogram into your to really profile your plant. We can also use the gaze chromatography, preferring for quantifying the volatile constituent, for instance, from your plant, such as essential oil that we know also are very important as a component of your traditional medicine. So in terms of really looking to the contamination of or adulteration, there are four important components that we need really to tackle when you are really dealing with that. Heavy metal toxicity. Some of the plant, some of the components can also be the mercury, the cadmium, the arsenic that you can also detect in most of African herbal concoction. This is very important. The component, the composition of those ingredients, but also the level, the proportion that are characterized them in your plant is very important to know. We also have to be very carefully when you are dealing with the traditional medicine. The microbial contamination is something that really is very important to consider because it can happen in your process. It can also be part of a material that you are using to really prepare your plant. So this is very important to know what are the total aerobic count for in terms of bacteria in your plant to know exactly, but you can use different method to do it. One of the most important that we also see in some of let's say the improved traditional medicine in our context, if an undeclared pharmaceutical product, most of the products that we have, medicine that we have in circulating in the West African region, for instance, are containing, for instance, some of the molecules which are not natural, but is a part of the product that people are really including there. If they are not natural, but they are boosting some activity in your in the traditional medicine. That sometimes is not what people are waiting for, but people are using like a second effect. For instance, we discover in some of our improved traditional medicine, the sildenafil as a part of a component. This is not natural. So we need to really be sure that all those components, part of your improved traditional medicine, are declared. We want to know exactly what is there, what type of component, what type of concentration, because some of the particles can be there, but the concentration is not enough to really make the toxicity. But some can be there, but even at low concentration, they can really be toxic for the organism. So in terms of really summary for quality control framework for medicine plan and the medicine delivery from them to ensure that that product are really meeting the quality, the safety, and the stability that are required, at least the minimum one, to be sure at least we can really put them in the market. So there should be good quality, safety, and we should also have a stability to be sure at least they are really not affecting the health of the user. So in terms of a regulatory landscape, we know in the continent, most of the countries in the continent are regulatory framework, but we know exactly that this is fragmented. We are not using the same SOP, so the regulatory is not harmonized across the different countries in Africa, for instance. And we also have some issue with post-market surveillance that are absent for some of the countries. Some of the clinical requirements are not really tackling in some of the countries. They are varying from country to country. We also notice that there is inconsistencies when you look when it comes to concept market authorization. Of course, we don't have a unified African pharmacopoeia for traditional medicine. This is very important. That's why we are saying that now having the African Medicine Agency coming on board, established now, really trying to get the continent country harmonized in terms of regulation framework can be one of the most important input in the area of traditional medicine. How to really unify the classification of traditional medicine, but also how to share some of pharmacovigilance data. We need to have AMAN need to work to get a very strong pharmacovigilance network that can really support the development of the improved traditional medicine in the continent. It's a big market, in the continent in terms of really the medicine market. This is more than $40 billion for medicine by 2030. So meaning that there is a way to really work to improve. Otherwise, we're getting in the market some of the products with a very poor quality or very bad regulation conducted in a different country. So there is also some gap that really we are seeing in a clinical area, for instance. The clinical trial, for instance, design for the polyherbal formula are not really much more utilized in a different country. We know there is also resistance for the practitioner in terms of formula disclosure, because some of the practitioners are really saying that we are not sure that what we have been to disclose as a formula for plant we are using will not be a take away, and we will not have any guarantee to be sure at least people will not be using those formula beyond them. So this is also some issue we need to work on that. We know there is a limited funding to really conduct phase 2, phase 3 clinical trial for the traditional medicine product. Even we want them to really be at the level where we can be sure that in terms of safety, everything is done to guarantee the use to the population. Of course, very challenging in methodological and standardized extract for the trial. We also notice that in terms of pharmacovigilance, there is a failure. There is no systematic adverse drug reporting for ITM in most of the country. We also notice that there is a drug interaction that are not documented. Most of the countries are using some traditional medicine. At the same time, they are using the commercial one, and that is sometimes quite difficult because we don't know yet what are the interaction between the natural product that people are really promoting, using, but also with the industrial commercial one now. So this is something that we really need to document it. We know that there are also some case of hepatotoxicity that can happen, but they are detected in the population and from the user. So we need also to work on those areas of pharmacovigilance to be sure at least we are really getting gathering most important data that can be useful for the safety of the population. So we also discussed there are 4 categories of the traditional medicine. For category 1 is in general that really are not subject to market requirement, authorization requirement. But for category 2, 3, and 4 should be really subject the market authorization. So depending of where you are, some of the people are decided, for instance, to really stay working on the category 1. Of course, there is an advantage, but also there is a disadvantage because you cannot move forward your traditional medicine to the population because you need to submit it to the regulatory framework. So when we try to really compare the 4 categories for traditional medicine, I'm saying that the category 1 don't need market authorization, but for the category 2, 3, and 4, they really need to be submitted to the regulatory authority to get approval before using them. In terms of preparation, we know that for category 1 and category 2 is based on traditional practitioner. But when we are dealing with category 3 and 4, meaning that we need to have licenses, facilities, your pharma industry should be there to really be sure that you are really following some of the important rules. In terms of production scale, for category 1 is base individual demanding, but for category 2, 3, and 4, this is semi-industrial. And for the active principle, for instance, is a crude raw material that is used for category 1 and category 2. But when we are moving to category 3 and 4, this is standardized extract or purified molecule. So for the GMP compliance, we don't need to have GMP for category 1, but the GMP is required and is mandatory for category 2, 3, and 4 when we are really dealing with that. So market authorization also, as I was saying, for category 2, 3, and 4, we need to really get this market authorization, submitting your dossier to regulatory authority, getting the feedback before really having your medicine or traditional medicine of a market. So this is very important. So you need to really submit the dossier depending on the country and the way and the content of composition of the dossier. And as I was saying, of course, we know that the regulatory framework is not well harmonized, but most of the countries has their own regulatory framework that we have to deal with that to be sure at least we are following what required to be have your category 2, 3, and 4 submitting to the market to the drug authority. The decision process is very important to be fulfilled. And we were saying that market authorization is mandatory for category 2 and 4 to 4. In general, the country is guaranteed 5 years validity and the possibility of renewal of your market authorization. That's what we are doing in the country in Burkina. But in case you are not respectful of those requirements, of course, we will have a suspension of your authorization, for instance. And sometimes it depends. You can also get some punishment because we are not really fulfilling the requirement that are being asked for the country. For Burkina, for instance, is a multi-minister enforcement. There are 4 ministers that are part of the process to be sure at least we are dealing with all aspects. The Minister of there, the Minister of Trading is there, the environment and the scientific research is also part of the ministers that are dealing with the process to really get your product on the market. For pharmacovigilance, I will say this is very important, and we need to also to have a very strong pharmacovigilance system to be sure at least we are protecting the population by using those traditional medicine. So sometimes it depends also of the area, because the genomic can be also used to really tackle the issue of authentication of ITM and also the surveillance. It will depend of what we need really to really assess. It can be used for quality assurance, for the safety surveillance, but also for the regulatory support. So in terms of really comparing some of the issue, the risk, and also the solution, if we don't have any quality control for your product we are using, of course, the risk will be focused on the value dosing, but also your efficacy will be questionable. So in terms of really a solution, we need really to working on a GMP basic reinforcement, but also using some of the reference standard. So this is very important. When you look at the fragmentation of regulatory, of course, we're getting on the market some unsafe product. So of course, we are really hoping that the African agency now will really work on harmonization on the continent, really strengthening the capacity of a different NRA in the different countries to be sure at least they can be dealing efficaciously with some of our product on the market. We also discuss the issue of pharmacovigilance. This is very important to be also considered in the framework of a regulatory aspect to be sure at least we'll be following all the side effects that can be happening when we are using the traditional medicine. So depending on the country where we are, the application for market authorization will depend on the local regulation. But the question is not who can apply. In general, the question is how to apply, and the component of the dossier. That really, I was saying, there is a fragmentation of regulation on some aspects, so it will depend from country to country. So in terms of a component, it will be differ, of course, between category 2 and category 3 and category 4, because the requirement will be different when we are really dealing with a different composition. So in terms of dossier, we have administrative dossier, we have a pharmaceutical dossier, the toxicology dossier, but also the clinical one when we are dealing with category 3. But when we are working on the category 4, what is very important is the labeling requirement. Including the requirement for category 3, we have to also to dealing with the labeling requirement for category 4. So in terms of renewal, I was saying that for most of the country, for Burkina, for instance, the guarantee, the validity of your market authorization is 5 years. You need to follow that. But if you are not really follow the requirement that are being stated by the country, of course, we can also face to some of the sanction, revocation for your market authorization, but we can also pay some fees for fine, moving from between $200 to $2,000. So in terms of take away message, I was saying that there is a risk proportionate framework for the category of product that we are really taking from improve traditional medicine, depending of the category that we have. But the category 2, 3, and 4 are very important, and they are to be following some of the requirements. We possibly demanding of clinical, but also toxicological evidence. We also discussed the fact that there is a very low documentation in some of the aspects, but also we need to have a standardization of some of the practice at the continent level, because we know that some of the aspects are not harmonized, but AMA is working on this way, and we are really hoping that at the end of the process, we have a very good harmonization of the process between the different countries in the continent. So those are the things that I want to share with you. Thank you very much. And I'm sorry by really losing the internet connection sometime. Thank you very much. So can you hear me? I have done. Can you hear me?
Yeah. Are you listening to me, Professor Isa Sulema?
Yes.
Thank you very much. I was saying, but maybe my background was muted, that this is the second activity between OIC Comtech and NRB Burkina Faso. The first activity was last month when OIC Comtech had conducted a training on GMP in your guidance in Ouagadougou. Now this is the second activity that you are taking as a speaker, as a resource person with OIC Comtech on quality assurance of medicine, herbal medicines with us. My question is, do you, ANRP, Ouagadougou, Burkina Faso, conduct clinical trials before recession of herbal medicines?
Yes, thank you very much for your question. Yes, for most of the ITM, we are conducting clinical trial before market authorization. And one of the very great examples is now there is a clinical trial ongoing of by using one of ITM in Burkina. We started just 1 week ago, and we'll be there for inspection to be sure that everything is also working well on the field. Before now, we submitted all the dossier to really get the market authorization. Yeah.
Okay, thank you, Dr. Sulaiman, for your presentation. Also, thank you for joining us. I would like to invite Dr. Abdul Rashid for the concluding remarks, if we have something to give to close this section.
I think just I finish very quickly. That's Assalamualaikum warahmatullahi wabarakatuh. Professor Dr. Rail from Jordan University, and also Dr. Tarim Sara, additional Dr. Quality Assurance and Lab Testing, and Dr. Isa Suleiman, ANRP Burkina Faso, and Dr. Farah and my colleagues, Roko. Very Assalamu alaikum wa rahmatullahi wa barakatuhu. As we come to the conclusion of the online training workshop on quality assurance and physicochemical analysis of drug formulations, I would like to express my sincere gratitude to all experts, speakers, which I have mentioned already, mentioned the names, organizer Roko, and participant who contributed for us on the success of this important webinar. Over the course of this webinar, we have explored key aspects of pharmaceutical quality assurance, analytical methodology, regulatory requirements, and best laboratory practices that are essential for ensuring the safety, efficacy, and quality of not only allopathic medicine, but also the herbal medicines. The knowledge shared by our distinguished experts and resource persons has highlighted the critical role of robust quality systems, quality assurance, quality control, and reliable physicochemical testing in protecting the public health and strengthening pharmaceutical industry across OIC countries, particularly the OIC African countries. Quality assurance is not merely a regulatory obligation, it is a commitment to patients and the society at large. Every medicine that reaches a patient must meet the highest standards of quality, safety, and effectiveness. In an era of increasing globalization of pharmaceutical manufacturing and supply chains, the need for harmonized standards, validated analytical methods, and competent human resources has become more significant than ever, even before. The discussion during this workshop have demonstrated that advances in analytical sciences and quality management system, that is QMS, provide powerful tools for ensuring quality pharmaceutical excellence. However, these tools can only be effective when supported by well-trained healthcare professionals, regulators, pharmacists, modern laboratories, strong regulatory frameworks, and a culture of continuous improvement. I am particularly encouraged by the active participation and engagement of attendees from various countries and institutions, particularly from DRAP, ANRP, and Jordan University. Such interaction promote the exchange of experiences, foster professional networks, and strengthen collaboration among scientists, regulators, quality assurance professionals, academia, and industry stakeholders. These partnerships are vital for addressing common challenges and advancing pharmaceutical quality standards throughout the OIC region. On behalf of OIC Comtech, I would like to reaffirm our commitment to supporting capacity building initiatives in pharmaceutical sciences, regulatory affairs, clinical research, herbal medicines, biotechnology, and healthcare innovation. We believe that investing in human resource development and scientific cooperation among OIC Muslim countries is fundamental to achieving self-reliance and sustainable growth in the health sector, particularly OIC 15 development reform and 57 Muslim countries. I encourage all participants to apply the knowledge and skill gained during this webinar in their respective organizations and institutions. Let us continue to promote a quality-driven culture, strengthen laboratory capabilities, enhance regulatory compliance, and contribute to the production of safe, effective, and high-quality medicines for our population with the rational use of drugs by the pharmacist with rational pricing. Before concluding, I would once again extend my heartfelt appreciation to the trainers for sharing their expertise, to the organizing team for their dedication and hard work, and to all participants for their enthusiasm and commitment to professional excellence. I hope this webinar will serve as a catalyst for future collaborations, research partnerships, and training opportunities among OIC 15 member states, particularly the West African countries. Together, we can strengthen pharmaceutical quality systems and contribute to better health outcomes for our communities. Thank you very much for your active participation and valuable contributions. I wish you continued success in our professional endeavors and look forward to meeting you again in the future of OIC Comtech program. Thank you very much. Assalamu alaikum wa rahmatullahi.
Thank you, Dr. Abdurrashid, for your remarks. Once again, I want to thank all our distinguished speakers, Dr. Arin, if she is still online, Dr. Isaka Sulama from Burkina Faso, and Dr. Tarim, thank you very much for joining us and share your expertise with you. Thank you for all participants for your engagement. Thank you very much.
Madam, thank
you.
You
have something.
मैं देता हूँ आप इस कुर्सी को साइड पे कर दे.
Okay.
Oh,
yeah.
You come. You are I understand. Go there. Kaka, you can take the photograph. Please come.